Hello students, this is Dr. Faizan Mirza. I'm discussing questions related to homeostatic control of excretion and the kidney function with you in this video. So here I'm taking this question from October November 2019, variant 42. This question basically asks about ADH hormone. ADH hormone is released into the blood of the mammal when changes occur in the internal environment. State one, change in the internal environment, which leads to release of ADH. We know when the water potential decrease or the ion concentration increase. This is a fact that you need to recall. So you can just state that. Name the part of the body that ADH is released from. Posterior pituitary. You don't just state pituitary. You have to state that it is released from posterior pituitary and not the anterior pituitary gland, because pituitary gland is having two parts, anterior and posterior. ADH is released from the posterior pituitary gland. Posterior means towards the back side of the body. It's anatomically located towards the back side of the brain. So shows figure 1.1 shows cells of one of the collecting ducts of the kidney. So we know the collecting duct cells are water impermeable and they must allow ADH to attach. So you can see what, let's just work on this figure first. So this is the membrane, and this must be a basolateral surface. Basolateral surface is the surface which is next to blood vessel. So the blood vessel must be here, somewhere here. So the blood vessel must be supplying blood from here, and the membrane here is having a receptor for ADH. Whereas the opposite surface, this is the apical surface. The apical surface of the cell will always face the lumen of the duct. We know that the entire nephron is made up of one cell thick region. So this is one cell thick region. On this side, you will have your blood. So this is your blood here, and here you will have your blood. In between, you will have your tissue fluid, and then you have this. This is the basolateral surface, as already mentioned to you. Then you have one cell thick cell of the nephron. Then here you have the apical surface, and this apical surface is facing the lumen, and this is where the fluid is traveling. The fluid which eventually is going to form urine. So this is how the fluids actually are there for every cell. Whenever we are discussing a nephron, you need to figure out which is the apical surface and which is the basal surface, and then work out accordingly. So ADH comes from the capillary, it binds to the receptor, it activates the enzyme phosphorylase. The phosphorylase enzyme allow these vesicles, which already have these stored proteins. We know that these proteins, these are aquaporin channels stored on the vesicle. This is the vesicle B, and these vesicles are already there. They start to move towards the membrane and they fuse. And when they fuse, they allow the aquaporin shells to get incorporated onto the apical surface. And when they got incorporated onto the apical surface, water starts to move into the cell, and this water then moves into the blood. So this is how water goes and gets absorbed by the ADH hormone across the collecting ducts. So many the membrane proteins A, and so the N and cell structure B. So this is A is receptor, B is vesicle. So this, I think this is very simple. The phosphorylase enzyme stimulates structure B. Describe the response of structure B to the stimulation and the consequences of this response. So you just need to describe the facts here. When ADH binds to a receptor on the basolateral surface, you need to make sure that you mention that the receptor is on the basolateral surface. The enzyme phosphorylase gets activated within the cytoplasm. Although I've not mentioned here, but it's worth mentioning that the enzyme gets activated within the cytoplasm. This stimulates the release of the structure of the structure B, not the release. The structure B, which are the vesicles of aquaporin channels, are stored in the cytoplasm, start moving towards the apical surface. These structures B are basically allow water channels to be incorporated into the apical surface. The cell becomes permeable to water, and it starts to move into the cell down the gradient from collecting there to the lumen. This allows more water to be reabsorbed in the blood and less water is lost in urine. So this basically covers the idea of how ADH is working. Now this question here is from Asian 2019 variant. This is from variant 42. Yes, this is variant 42. So this question came, and this question basically was a photomicrograph of a cortex of a kidney. So whenever you are seeing for cortex of a kidney, this is structure, this C-shaped structure that you see actually is your capsular space. This is the capsule space, and this entire something like this region is basically your Bowman's capsule. And this Bowman's capsule, any point within the Bowman's capsule is basically the lumen of the Bowman's capsule and the renal capsule. At times they ask you to label the podocyte cell, although not asked here. So podocyte cell, where is podocyte cell present? So the podocyte cell will be present on this region of the Bowman's capsule. So you can just, if they ask you for the podocyte cell, so for podocyte cell, you will just mention that this is where the podocyte cells are present. If they ask, you will not mention this. This is an incorrect answer for podocyte cells. Similarly, the blank region here will be a wrong answer for podocyte cell. The podocyte cells are present only here. The lumen, since they are asking lumen, so you'll just mention anywhere in the blank. So that's the lumen L. Glomerulus, this bunch of capillaries, this entirely is the glomerulus. This all is the, you can see bunch of capillaries, the blood vessels here. So this is the glomerulus. You just label where the glomerulus is. They're asking you that the mass greater than 68,000 are prevented from passing the Bowman's capsule. Name the structure that acts the filtration barrier. So basement membrane. Basement membrane is always the structure that prevents which serves as the filtration barrier. The size barrier also is the filtration sets and the fenestration. But yes, the basement membrane is the actual structure which prevents the substances larger than that mass to be passing into the filtrate. Now they're saying that how the glomerular filtrate can change. We know if you just read through the information, they are saying GFR is a flow rate of the fluid from in the kidneys per unit time. Efferent arteriole supply blood to glomerulus of each nephron of the kidney, while efferent arteriole take the blood away from each glomerular. The lumen appears of the lumen can change. So we know that this is the, for example, your Bowman's capsule. It is receiving from blood from the efferent arteriole. The blood goes ahead into the efferent arteriole. What already you know that efferent arteriole is wider and the efferent arteriole is narrower. When efferent arteriole is narrow, I'm using E as efferent and A as efferent. So when efferent arteriole is narrower, it allows more blood to go and get filtered, and the blood pressure in this region, in the glomerulus, this region will have a higher blood pressure, so more blood gets filtered out. So they are saying that how the lumen diameter changes will be assisted. So if the efferent diameter is normal and efferent is normal, the GFR is normal. But if the efferent arteriole diameter is decreased, and the efferent arteriole lumen diameter increases, the GFR would decrease because the blood can actually go into the efferent at a very fast pace, so less of filtration will occur. Then they are saying the lumen of arteriole efferent stays normal, whereas efferent increase, again the GFR will decrease because again for the same sake that in the efferent the blood can go faster, it will be spending less time in the glomerulus, so the blood pressure in the glomerulus will be lower. After learning, they are asking you to describe between how the glucose and glomerular filtrate can be reabsorbed back into the blood. So you need to just recall how PCT absorbs it. So you can mention tight junctions here. You can mention the apical surface and the basal surface. They have micro lab brush border. And you can mention how the sodium potassium pump is present on the basolateral surface that keeps the sodium concentration low within the PCT cell, which is the driving force for the co-transporter protein present on the apical surface. And this co-transporter protein allows sodium to move together with glucose or with amino acid. Here they're asking about glucose. So you just mentioned sodium and glucose being transported by co-transporter proteins on the apical surface into the PCT cell. Once in the PCT cell, it goes into the blood. So this drag you need to just explain. And the mitochondria, yes, you need to explain mitochondria here. That mitochondria, they provide ATP for the functioning of the sodium potassium pump, and the tight junctions, they prevent the tight junctions prevent the free mixing of this filtrate. And this will cover 5 marks. Low concentration of sodium is important for driving force for the co-transporter protein to function. The placement of the proteins, where sodium potassium pump is present. Sodium potassium pump is present on the basal surface, and the co-transporter proteins is present on the apical surface. They must be mentioned clearly. Etienne explained 3 features of proximal tubule that help it in function. So it's having many mitochondria that provide ATP for carrying out the active transport, and sodium ions and basolateral surface. Tight junction that will prevent the mixing of the filtrate. Tissue move from microvilli brush border on the apical surface. So that will give you good marks. I hope these simple questions were helpful for you. That's it from my side. Thank you so much.